Hello there everyone. As we're waiting for Benito to arrive, what better thing to do than to watch this wonderful feline specimen? Majestic!
Hello everyone and welcome. It is the second video in my series of acquisition of behaviour. Now first, if you think I sound a bit funny, that's because I've got a killer of a sore throat at the moment. But don't worry, I'm pulling out all the stops. Look at all these layers I've got on. There's no way this cold is going to approach me. I shall defeat it, believe me. So, today we're going to carry on kind of from where we left off last time. Remember last time we talked about how behaviour can be innate or learnt. Is it innate or learnt, or is it a mixture of the two? And today we're going to focus more on the role of genetics on behaviour. Now on the diagram here, I've got a very basic diagram here. I'm sorry, it is very small, but don't worry, I'm going to read everything out for you. So we've got genes, genetics, and the environment coming together, just like how we talked about last time, how no behaviour is strictly innate or is strictly learned. It's a mixture of the two, so the genes and the environment coming together, that leads to some cognitive mechanisms within the animal, and that is what produces your behaviour. That produces an outcome, and as a result, possibly a fitness advantage. So, if there is a fitness advantage, that gene which caused that fitness advantage will be selected for by natural selection. If it's not, it will be lost through gene attrition. Okay? So this is sort of basic Darwinian natural selection. However, we learned in the last video that a genetic trait such as birdsong can be learnt over time. But an example of where a behaviour does have a strict genetic basis is we're showing a study on blackcaps. Now, blackcaps, beautiful birds, part of the warbler family. There are various species and subspecies across Europe, and some are migratory and some are not. So the species that you get in Germany, that you find in Germany, are migratory, and ones that you find in Cape Verde are not. What these scientists did is that they measured migratory behaviour by looking at the amount of restlessness that the birds have at the time they need to migrate. So they kept birds and measured how restless they were. The idea is that restless birds are the migratory ones, so the ones that want to move about, go away and move somewhere else, so they're more restless, whilst the Cape Verde birds are more content where they are. They're happy with life as it is. So what they did, they purebred the two subspecies and found that the non-migratory ones were non-restless and the migratory ones were restless. They then crossed the two subspecies to produce hybrids. So, will the hybrids be restless or non-restless, or a mixture of the two? Well, it was found that these hybrids were actually restless. So they were just as restless as the purebred migratory birds. So this is pointing fingers towards this migratory behaviour having some sort of genetic component involved. Another example is in two species of tree frog, the grey tree frog and the pine woods tree frog. And these two different tree frogs have different calls which they use to attract females. That's what the males do to attract females. So once again, they cross them to produce hybrids, and they found that the hybrid call was sort of an intermediate between the two parent species calls. They then tested for female preference towards different calls, and they found that hybrid females preferred the calls of hybrid males. So both these results indicate even further that there is a genetic basis to the behaviour that we see. Now, in an ideal world, we want to observe behaviour in a natural environment, but also in an experimental way, so we can see which ones survive and which ones don't. So we want it to be in a natural environment, but in a way that we can control what's going on. Now, that's very difficult in nature, as you might imagine. Especially when you're looking at smaller things. I mean, they're a real nightmare to look at. When looking at things, let's go away from animals now, when looking at yeast, for example, you can only really look at them in a petri dish, bless them, but that's a shame because yeast cells show this, what's called the snowflake phenotype, where under stressful conditions they sort of clump together. And there are theories stating that this could have led to the origin of multicellularity, which is where we are today. Well, hey, here I am, hello. But it's very difficult to observe this sort of behaviour outside the lab, so it's difficult to get good results, basically. From a behavioural ecologist perspective, we can study this behaviour in the genetics of behaviour, blah blah blah, using two methods, basically. The first one being observation and experiment, which identifies the present function of a behaviour, and there's also a comparative method, so it compares the function of this behaviour under different selection pressures or between different species, etc. Now, a good example of an observation experiment was conducted by Tinbergen, the main man that we talked about last time when there's 4 Ys. And he looked at black-headed gulls, which is probably one of the most common gull species you can find in the UK, so he didn't exactly push himself, did he? No offence, Tinbergen. But anyway, so these are ground-nesting birds which lay eggs. Obviously all birds lay eggs, what am I talking about? But he noticed that, like many birds, these black-headed gulls, once the chicks hatch, they take the shells and deposit them somewhere else. And Tinbergen hypothesised that they were doing this to make the nest less conspicuous to predators which would eat the chicks. So things like herring gulls, carrion crows, things like that. So what he did, he looked at 120 nests, and for 60 of them, well, half of them, he got the shells back and placed them back near the nest. And he showed that nests which have the shells near them are more conspicuous because the inside of an eggshell is really a real bright white colour. It acts as a flag to predators saying, come over here, there's food over here. So the behaviour of removing the eggshell and depositing somewhere else is a strongly naturally, positively selected trait. However, this study was kind of criticised because Tinbergen didn't actually measure selection directly. But never mind, Tinbergen, he's awesome, he can do what he wants, can't he? So, back in Tinbergen's day, you'd usually observe a behaviour in the field, design a hypothesis, then create an experimental design, conduct your experiment, and then see if it matches your hypothesis. Observation in designing experiments is less normal nowadays. Scientists nowadays rely much more on mathematical modelling and all different types of modelling to design their experiments. So it's become a whole lot more technological. So using this mathematical modelling, we can build specific hypotheses from a comparative pattern. So this brings us on to our second method. But another thing which goes against observation is that it's very difficult to control everything. Ideally, you would want to control all the other things which could be changing, so I don't know, obvious things like temperature and whatnot. So you'd want all of them to be constant, except the trait that you're measuring. And in nature, that's bloody difficult to do, so it's very difficult to get good results when studying behaviour. So only then you can be really sure that you've got natural selection acting on a trait. Okay, so the comparative method helps us compare associations between traits. And the logic is that shared traits have this, between different species have the same adaptive value. And these associations can be made with species which are closely related or distantly related. Now, a good example is in ungulates. So species of ungulate which live in vast open spaces tend to live in large groups or herds in some cases. Okay, whilst other species which prefer to live in dense woodland like to live solitary lifestyles. So is the evolution of group size based on habitat use, for an example? Another example is dimorphism in primates. So in species of primate where the female only mates with one male, but that male is free to do whatever he likes, he can mate with many females. In those cases, the male seems to be drastically different from the female. Whereas in monogamous species, the male and female seem to be incredibly similar. So this difference in appearance of males and females could be due to a whole load of things. Competition, fighting off other males, impressing the female, with elaborate traits, etc., etc. However, ultimately, all primates have a common ancestor, so it could just be that the common ancestor had that specific trait, and that's just inadvertently, if you like, been passed on to its descendants. So there may not be an adaptive function at all. So when doing experiments like this, you must always control for phylogeny to see whether a particular trait has evolved multiple times within a group and whether it actually has an adaptive function. And of course it's also important to do this over multiple generations, like most of the experiments that we're trying to do here when it comes to the genetics of behaviour. Because otherwise we could just be getting correlations and not actually proper cause and effect type relationships. Because remember, correlation does not mean causation. And I couldn't really do a video on what causes behaviour without talking about the brain, could I? Because it is the epicentre of all behavioural regulations. But it's actually surprisingly difficult to use the comparative method when looking at brain size, because obviously brain size also increases with body size. Okay, so instead, if you plot body size on the x-axis and brain size on the y-axis, and then you look at the residuals to your regression line, these residuals give us more useful information on how the brain controls behaviour. And I've listed here a load of things which it controls, and some of these might surprise you a little bit. So let's go through them. So reproductive behaviour. Species which pair bond for life, for example, tend to have larger brains. It's also been linked to innovatory behaviour, migratory behaviour. Birds that migrate have smaller brains than ones, well comparatively, than ones which stay where they are. And the reason behind that is that the ones that stay where they are have to cope with harsher conditions. So when you think of like swallows which bugger off to Africa for the summer, they don't have to endure the harsh wintry British winter. So being a non-migratory bird with a larger brain gives you a greater capacity to think outside the box, if you like, and find new foods in an otherwise harsh environment. It's also been linked to longevity, so birds which live longer tend to have larger brains, and this has been linked to be because of extra cognitive abilities to cope with environmental stress, so they're less likely to die. Also linked to invasion success, so invasive species. The success of an invasive species, you can actually predict that by looking at the size of the animal's brain, and you can make predictions as to how successful they would be at invading an area. And lastly, food storing behaviour. So you may know that many birds, mammals, all kinds of things cache food for later, and it's shown that by looking at the residuals again, animals that do this have a larger brain, particularly the volume of their hippocampus is larger. And obviously this is looking at differences in behaviour between species. But what about within species? Are there intraspecific differences in behaviour? Well, the answer is yes, particularly in social insects. Now, a guy called O'Donnell studied the paper wasp. And obviously, in the colony of social insects, you've got queens, you've got workers, but the two different morphs, although they're the same species, do very different jobs. So O'Donnell looked at the brain size of these different morphs, looked at particular regions of the brain, and they showed that the brains of the workers had more enhanced areas to do with visual stimuli, because they're out and about more, so they want to respond more to visual stimuli. When looking at regions in the brain of queens, on the other hand, they found there are enhanced regions involved in antennal processing, that sort of thing. Now, pure genetic determination of behaviour is rare, even more so if it is determined by only one gene. But this has been found. There are some examples, and one example is in Drosophila melanogaster, a fruit fly, probably one of the most studied animals on the planet. So if we were going to find an example, it would be in Drosophila melanogaster, and it is this 4 gene. That's the name of the gene, it's called 4. And that stands for foraging, believe it or not. So it's all to do with their foraging behaviour. Now, there are 2 different alleles here. We've got the 4R, and the R there stands for rovers. These rovers, they're called rovers because they move about a lot, but eat very little. On the other hand, you've got the complete opposite. You've got the 4S, which are the sitters, which move very little, but eat a lot. Okay, so depending on what allele you have, it depends what foraging behaviour you have. Now, obviously, usually it isn't just one gene, it's usually complex, a whole collection of genes which can influence behaviour sometimes. An example of this is the GP9 supergene complex in fire ants. Now, fire ants are social insects, they form colonies, and the species is divided into 2 social morphs. So there you have colonies which only accept one queen, so in one colony there's only one queen, and you also get colonies which have multiple queens. Okay, and depending what colony you are depends on the alleles you have in this supergene complex, which contains the GP9 gene. So there's allelic variations, so you have either uppercase B, homozygotes, or heterozygotes, so big B, little b. Okay? So if you have a colony which only has big B, homozygote, workers, then that's a single queen colony. They'll only accept one queen in that colony, and they'll kill any other intruders. On the other hand, if you have a colony which has homozygous big B or heterozygous workers, then they'll accept multiple queens. Now there could be a possible advantage of this, because queens which live in multiple queen colonies tend to be a lot smaller and have lower fat reserves. Remember, they're the same species, but it's just these subtle differences. Now, if we jump back to our example with Drosophila melanogaster, this foraging behaviour isn't completely genetically determined. It does allow for a little bit of plasticity, and this is shown in an experiment where these 2 morphs were starved. Okay, so the sitters, which eat a lot, were starved. It didn't really have much of an effect on the rovers because they don't eat very much anyway. Okay, so when the sitters were starved, they began to behave a little bit more like the rovers. So they showed some plasticity in their behaviour. So they can alter the way they behave depending on their environment, their environmental situation. And you can see the fitness benefit of that. If you're just stuck to doing one thing, then if your environment changes, then you're going to be pretty buggered, quite frankly. So it's important, yes, some behaviour is genetically determined, but it's good to have that plasticity, so if the environment does change around you, you'll be able to cope with it fine. Another example of this phenotypic plasticity was done in a study in honeybees. Now, we've already talked about social insects a lot in this video. Of course, honeybees are social insects. And we've talked about the different morphs you get. Now, within the workers, you get nurses and foragers. So obviously nurses stay in the colony looking after the larvae, etc., whilst the foragers are out looking for food to bring back to the nest. Now, what kind of worker you are has been shown to be dependent on your gene expression. So you have this differential gene expression, and usually bees become nurses to start with and then develop into foragers. So you get this change in gene expression. But it's also been shown that this doesn't just have to happen with age. If the bee colony, for example, is put under threat, so a load of foragers are killed by some mysterious flying monster or something, then the nurser bees can switch to becoming foragers and then helping maintain the stability of the colony. So it's very useful having this plasticity. You can see it's very, very important to be plastic. And what causes this differential gene expression is likely to be due to epigenetics, so DNA methylation and all that kind of stuff. A load of chemistry, basically, that we don't really need to go into. But that's all I've got time for today. I hope you enjoyed that. Quite interesting. Next time we'll be looking at the fundamental mechanisms of behaviour, starting with perception, so that should be good. But, oh god, I'm going to have some, I don't know what I'm going to have. Something to cure my throat. Oh, please help me. Any top tips, anyone? But anyway, I'm sure it'll be fine, really. I mean, there's nothing, my voice has been fine. I don't know what